Welcome back to this channel. Greetings to all. Ensure that you subscribe so that whenever a video is posted, you can get a notification to watch and learn whatever is detailed in that video. So today we're going to look at the structure of the human cell. And when you look at the structure of the human cell, it is composed of various parts. We have our nucleus inside here. This is our nucleus, which has the nucleolus. Then we have the cytoplasm. We have the cytoplasm, which contains various elements. We have the Golgi apparatus, we have the Golgi vesicles, we have the lysosome, we have the rough endoplasmic reticulum, smooth endoplasmic reticulum, we have the microtubules, we have the mitochondria, of course, not forgetting the ribosomes. So those are the parts of the human cell. So each part of the human cell has its own function. So to begin with, you're going to start with the nucleus. So the nucleus, it's contained within a nuclear, of course, within a nuclear envelope, which we can also call the membrane. And it contains, we must appreciate that the nucleus contains the DNA, which is 95% protein in nature. And in terms of diagnosis, it is essential because its staining characteristics aids in diagnosis of abnormalities and malignancies. Then the stored DNA, to give more details, determines the characteristics of cell, of course through synthesis of ribosomal RNA, messenger RNA, and transfer RNA. So it also controls the cell reproduction. So major parts of the nucleus, when you look at the nucleus itself, it has various structures. One is the nucleolus, nucleolus, and nucleolus produces most of the ribosomal RNA that aids in formation of proteins. Then number two, as far as the nucleus is concerned, we have the nuclear, it has the nuclear membrane. Of course, it is an envelope or limiting membrane that is composed of two unit membranes. So these membranes periodically come together to form what we call a nuclear pore. So the other part which is very important, of course, from the nuclear membrane, we have said we are reaching the nuclear pores, and of course the nuclear pores act as avenues for communication between the nucleus and the cytoplasm. The other structure within the nucleus, we have the chromatin, and we must appreciate that the chromatin is a thread-like material, of course made up of large molecules of DNA. And it is very important for packaging DNA into small volume to fit into the nucleus of the cell, and of course it also protects the DNA structure. So that is the chromatin. So apart from the nucleus, as we have mentioned, it has the nucleus, it has the nucleolus, the nuclear membrane, from where we get the nuclear pores, which is very important in communication between the nucleus and the cytoplasm, then the chromatin. So going forward, the other part of the human cell is the cytoplasm. And when you look at the cytoplasm, is a fluid into which other intracellular organelles bathe for nourishment. And of course it is more basic, it's more of basic components, hence having high affinity for acidic dyes like eosin. And of course it is within the cytoplasm that most of the work ordered by the nucleus is carried out. And the cytoplasm is made up of various organelles. One of them is the mitochondria. Or what can be called mitochondrion, which is cigar-shaped. When you look at its structure, the mitochondria is cigar-shaped, sometimes called ellipsoidal, and it occurs in various numbers and sizes. So the number of mitochondria vary from cell to cell, with the highest number found in cells that have highest metabolic activity, for example, the hepatocytes. So we must appreciate the major function of the mitochondria, and the major function is that are the energy producing powerhouse of the cell. So mitochondria basically, of course, as we have seen, this is mitochondria, this is mitochondria. So the number of the mitochondria, as you have said earlier on, varies from one cell to another, basing on the metabolic rate. And to give an example of hepatocytes, those are liver cells, whereby they contain up to 2,000 mitochondria. So the function of the mitochondrion are the energy producing powerhouse of the cell. So all the functions within the cell require energy. So the mitochondria is very important in ensuring that there is energy produced. So it's also the site by which food energy is made available for metabolism of the cell. So apart from the powerhouse, the mitochondria being the powerhouse of the cell, it also performs other functions. For example, regulation. It's very important in regulation of apoptosis. So apoptosis is programmed cell death, whereby once the cells have reached their lifespan, they must be removed from the body, and that process of removal of old cells or damaged cells is known as apoptosis. So the second organelle within the cytoplasm are the ribosomes. And one peculiar feature of the ribosomes is that they are non-membrane bound cytoplasmic organelles. Very important statement. And the ribosomes are basically involved in protein synthesis. So protein synthesis occurs within the ribosomes. So we must appreciate that they also the ribosomes act as assembly line where RNA from the nucleus is used to synthesize proteins from the amino acids. So, of course, the ribosomes are made up of two subunits. We have the large and small subunits. And of course, when you look at the eukaryotic cell, eukaryotic cell ribosomes is of the 80S ribosome, which is made up of 60S large subunit and the 40S small subunit. The third organelle of the cytoplasm, or the third organelle found in the cytoplasm, is the endoplasmic reticulum. And of course, the endoplasmic reticulum is a series of membrane-bound channels that are basically pathways. They are basically pathways for transportation of secretory products that are either used by the cell or released out of the cell. And of course, the endoplasmic reticulum is of two kinds. We have the rough endoplasmic reticulum, which you call also the granular endoplasmic reticulum. Then we have the smooth endoplasmic reticulum, which you call the agranular. The agranular endoplasmic reticulum. So basically, the reticulum, as we have said, is divided or is of two kinds: the rough endoplasmic and the smooth endoplasmic. So the granular endoplasmic reticulum, or the rough endoplasmic reticulum, of course, is abundant in cells that actively produce secretions, or what we call the proteins. So that's the major function of endoplasmic reticulum. And of course, this secretion of proteins may be released outside the cell, and of course they have high affinity for the basic dyes. Apart from that, we must appreciate that the granular endoplasmic reticulum is a major site for protein synthesis, Number 2, it's a site or assembly site for the phospholipids, and also conveyor channel for transport of secretory products to the Golgi complex for packaging. Then the smooth endoplasmic reticulum, or what we call the agranular endoplasmic reticulum, generally is involved one in detoxification of both endogenous and exogenous toxic substances. Endogenous, these are generated within the body, and exogenous, they come out of the body. Number 2, the agranular endoplasmic reticulum is involved in synthesis of steroid hormones. Number 3 is very important in lipid membrane, of course, lipid synthesis, membrane synthesis, and repair. Then number 4, it's very important in release of calcium ions in myocytes during muscle contraction and relaxation. And finally, a granular endoplasmic reticulum acts as accumulation of absorbed fatty acids in the ileum prior to transition to the Golgi body. The fourth organelle within the cytoplasm is the Golgi apparatus. So when you look at the Golgi apparatus, of course, it's a network. The Golgi apparatus is a network of reticulum comprising of the smooth surfaced cisternae, which you can also call a lamellae with lumen vesicles and the vacuoles. So, of course, the Golgi apparatus has two faces. We have the cis face and the trans. We have the cis face and the trans face. So the cis face is where the nucleus sits. We must appreciate that. And looking at the major function of the Golgi apparatus, we must appreciate that once ribosomes have synthesized the proteins, these proteins are delivered by the endoplasmic reticulum. However, they are concentrated and then packaged in the Golgi vesicles. Of course, one of the parts of the Golgi apparatus is the Golgi vesicle, or what we call the secretory vesicles. And once they are packaged in the Golgi vesicles, they are able to be transported. They can now be transported to the cell surface. So apart from that key function of the Golgi apparatus, the other functions include formation of lysosomes and other microbodies like the peroxisomes. Also, the Golgi apparatus is very important in formation of acrosome of the spermatozoan head. Also, the Golgi apparatus acts as an assembly of large fat macromolecules such as the phospholipids. Then we must appreciate that the Golgi apparatus is a site for synthesis of glycoproteins. So that is the Golgi apparatus. So the fifth organelle within the cytoplasm are the lysosomes. So the lysosomes, of course, they are dense membrane-bound organelles. Must appreciate that. And they majorly contain hydrolytic enzymes. And these hydrolytic enzymes act in acidic pH. And because they act in acidic pH, they can also be called, or they are named, acid hydrolases. Examples of these enzymes include acid phosphatase. We can have the acid ribonuclease and others. So when you look at the lysosome, of course, it is made up of a membrane, as we have said. Of course, it has the enzymes, very important, and composed of also the lipid bilayer, and also it has what we call the glycosylated membrane transport proteins. So when you look at the structure of the lysosome, you should be able to appreciate that. So basically, when you look at the lysosomes, under no stimulation, the enzymes are safely inert, because we have said it contains hydrolytic enzymes. So when there is no stimulation, these enzymes are inert, bound to the lysosome membrane, because we have said it has a membrane. However, in case there is membrane breakage, these enzymes can leak into the cytoplasm, and when they leak into the cytoplasm, they cause what we call self-digestion. They digest away cytoplasmic organelles. Okay, and of course, when they digest the cytoplasmic organelles, they lead to what we call cell lysis or cell autolysis. So basically, when you look at the function of the lysosomes in general, they are basically involved in breakdown. They're involved in breakdown of particles engulfed by the phagocytes. For example, the neutrophils and macrophages, such as infecting viruses and bacteria, the worn-out organelles, food particles, and others. So, lysosomes, when you look at the lysosomes, lysosomes. They are involved in cell digestion in sites of disease and injury. Then number 2, during programmed cell death. And number 3, deterioration of mammary glands, and also phagocytosis. Phagocytosis as carried out by the neutrophils, macrophages. So the other key aspect we must appreciate about the lysosomes is that they're involved in removal of damaged organelles. So because of these functions of the lysosomes, they are also, or they are named as the digestive system key point. They are also referred to as the digestive system of the cell. So that is about the lysosomes. Then the other key aspect we should be able to appreciate about the cytoplasm in far as organelles are concerned, the cytoskeleton. Cytoskeleton. So when you look at the cytoskeleton, of course, they are made up of 3 kinds of protein. The protein filaments, and one is the actin filaments, which can also be called the microfilaments. Number 2, we have the intermediate filaments, and number 3, we have the microtubules. So those are the 3 kinds of protein filaments that are contained within the cytoskeleton. So we also need to appreciate the functions of the cytoskeleton. Functions, when you look at the functions of the cytoskeleton, the cytoskeleton, which we have said is made up of 3 kinds of protein filaments, the actin, intermediate, and microtubules. The cytoskeleton provides what we call mechanical strength to the cell. And number 2, they're important in chromosomal separation. As we appreciated the cell cycle, we are going to appreciate in cell cycle how the cell divides to form the daughter cells. So the cytoskeleton is involved in chromosomal separation in mitosis and meiosis. Then also it's very important in intracellular transport of organelles. The other aspect is that the cytoskeleton anchors. They anchor organelles in place, and also they are involved in cell locomotion. So those are some of the functions of the cytoskeleton. Then, of course, we should be able to appreciate. We talked about the peroxisomes, but basically, when we look at the peroxisomes as micro bodies, just to highlight something about them, the peroxisomes, they are majorly micro bodies. bound organelles resembling lysosomes, but are different in that they contain a set of enzymes called oxidases. So that's basically about the peroxisomes, and these oxidases can include the catalase, D-amino oxidase, and others. And of course, these oxidases, they play a key role in what we call oxidation and get rid of cell toxic peroxidases or potentially toxic metabolites such as phenol and alcohol. So you're going to find that peroxisomes are abundant in the liver and kidney, where major detoxification occurs. The other key part we need to appreciate is the plasma membrane. Very important. You can put it as number 7. The plasma membrane, which sometimes is called the plasma lemma. So one thing we need to appreciate about the plasma membrane is that it's a double layer of lipids and proteins that surrounds a cell and separates the cytoplasm from its surrounding environment. So when you look at the plasma membrane or the plasma lemma is made up of proteins, phospholipids, glycolipids, cholesterol, and glycolipids. So when we talk about the plasma membrane, we should appreciate the proteins, phospholipids, glycolipids, cholesterol, and phospholipids. So we're going to talk about each in brief. Of course, looking at the phospholipids. As we have said, it is one of the components of the plasma membrane. Is that the plasma membrane is made up of 2 layers of phospholipids. So we have 2 layers of phospholipids, which are arranged to form a double-layered sheet that we call the phospholipid bilayer. So what is the key function of this phospholipid bilayer? So the bilayer membrane is structured in a very unique manner. One with what we call hydrophobic tails, or the non-polar tails positioned together inside. And number 2, it is made up of hydrophilic, what we call the polar heads, which are positioned outside of the bilayer sheet. So the hydrophobic tails and the hydrophobic are very important in that this unique structure, together with the integral transporter proteins, regulate the permeability of materials through the membranes. So that is key about the lipid bilayer. So When you talk about the polar heads, we should also appreciate that they are water loving. The polar heads, they are water loving or liking, and of course they are highly attracted to water because they are water loving. Then the non-polar tails, when we talk about the non-polar tails, we're looking at the hydrophobic tails. They are water hating. So when we talk about the non-polar tails, for them they are water hating, and because they are water hating or fearing, they are not attracted to water, but they are attracted to highly permeable to what we call oily molecules. For example, the fatty acids. Cholesterol and this cholesterol is very important in maintaining what we call fluidity, and also increases the stability of the membrane. So without cholesterol, the membrane would easily split apart. Then the other key part of the plasma membrane is the glycoproteins, and these glycoproteins, they serve as attachment for other cells to help in recognition of an interaction with other cells. Number 2, they serve as attachment for infectious pathogens, for example, bacteria and viruses. And of course, they act as attachment for toxins, hormones, nutrients, and other molecules. Then the other component of the plasma membrane, they are glycolipids. These glycolipids majorly serve as antigens. They serve as antigens for body's cell-to-cell recognition. In other words, we're talking about the ability of a cell to distinguish one cell from another, such as in the ABO blood system. Then, of course, we'll talk about the proteins as a component of the plasma membrane. So some membrane proteins, one, act as transport proteins. And these transport proteins that channel substances into or out of the cell. For example, we have the integral and integral or intrinsic proteins. That's about the proteins. Then, of course, the plasma membrane, as we have said, made up of the phospholipids, the membrane cholesterol, glycoproteins, glycolipids, and the proteins. As we are concluding with the proteins, in as far as the plasma membrane is concerned, we must also appreciate that several models describe the arrangement of proteins and lipids in the plasma membrane. For example, we have the lipid bilayer model originally proposed for membrane structure. and this was done by Davison and Daniel in 1935. However, apart from that, the fluid mosaic model, the fluid mosaic model, which was proposed by Singer and Nicholson, is the more recent and acceptable model for membrane structure. So, what's all about fluid mosaic model? So it's called fluid because individual phospholipids and proteins can move around freely within the layer like it's a liquid. So that's why it is called fluid. Then of course mosaic. We want to talk about mosaic because of the pattern produced by the scattered proteins molecules when the membrane is viewed from above. So as far as Singer and Nicholson fluid mosaic model is concerned, they stated that the plasma membrane is essentially composed of a lipid bilayer. That's one. And of course, the hydrophobic, the nonpolar region, or the tails of the lipid face each other at the core of the bilayer, while the hydrophilic regions face outward. Then number two, according to Singer and Nicholson, the globular proteins are irregularly embedded in the lipid bilayer. Then number three, the membrane proteins are arranged in different ways, whereby we have the extrinsic or the peripheral membrane proteins that are loosely held to the surface of the membrane, and they can be easily separated. For example, the cytochrome of the mitochondria. Then we have the intrinsic, or what we can call the integral membrane proteins, which are tightly bound to the lipid bilayer, and they can be separated only by use of detergents or organic solvents like the hormone receptors, cytochrome, P450, and others. So that's about the fluid mosaic model. But generally, the cell membrane plays the following function. One, acts as a selective permeable barrier to molecules. So not all molecules must enter the cell, so it is very selective which molecule should pass across the membrane. Number two, according to its structure, it keeps the cell contents together. That's another general function of the cell membrane. Then of course, not forgetting that it's very important in carrying out communication with other cells. Number four is very important in recognition of other external substances, e.g. in ABO blood group system. Then number five is the site of various chemical reactions, e.g. it controls the passage of substances in and out of the cell. So as we conclude the structure of the cell, we also must appreciate that there are some substructures or elements of the cell that can be seen by light and electron microscope, and there are some which are only seen by electron microscope. For example, when you look at the nucleus, cell nucleus, cytoplasm, the chromatin, rough endoplasmic reticulum, nucleolus, and cell membrane, they can be seen by both light and electron microscope. However, the nuclear membrane, nuclear pores, mitochondria, ribosomes, Golgi apparatus, centrioles, and the lysosomes are basically seen by electron microscope. Thank you for listening.